The overall goal of this procedure is to generate Illumina mRNA sequence libraries from small amounts of starting material. The first step of the procedure is to reverse transcribe total RNA with DT-T7 primers to produce cDNA. The cDNA is used for RNA amplification by in vitro transcription. Subsequently, the amplified RNA is subjected to random fragmentation. Reverse transcription of the fragmented RNA produces cDNA, which undergoes adapter ligation, size selection, and PCR amplification. Ultimately, this protocol shows the gene expression profile of the sample through measurement of gene copy numbers.
The main advantage of this technique is that it requires only 10 nanograms of starting total RNA, that is, orders of magnitude less than what is required from other existing protocols. Moreover, it can generate paired-end libraries from such small starting RNA quantities.
This method will allow scientists to explore novel questions in biology by allowing the determination of gene expression profiles from difficult-to-obtain cell populations, such as those derived from laser capture microdissection or from flow cytometry.
We first had the idea developed in this method when we needed to profile gene expression from a very limited number of cells differentiated from human embryonic stem cells.
Visual demonstration of this method is critical, as the RNA handling steps are difficult to learn due to the very small amount of RNA that can be easily degraded.
Demonstrating the procedure will be Angela Elwell, Jennifer Bolin, and Bao Kim Nguyen from my laboratory.